Hey everyone! Welcome back to our channel where we bring you the latest and greatest in health science topics. And today we're diving into the world of clinical trials. When a new treatment, medication, or intervention is introduced in medicine, it must be rigorously evaluated to understand its effectiveness and safety. For this purpose, we use clinical trials. These experimental studies are crucial for making evidence-based decisions and improving patient outcomes. In this class, we will break down what clinical trials are, how they are used, the types of trials that exist, key methods such as randomization, blinding, concealment, and matching, and how these methods influence the statistics of the results. So, let's dive right in. First up, let's discuss what a clinical trial actually is. A clinical trial is a type of research in which an intervention is tested on a group of people to evaluate its effect on a disease, condition, or health event. This may involve new drugs, surgical procedures, non-pharmacological treatments, or medical devices. The primary purpose of clinical trials is to compare different treatments or strategies to find the most effective or safest option. So, why are clinical trials important? Clinical trials are essential for several reasons. They help us determine the effectiveness and safety of new treatments. They compare different treatments to establish which ones are better or safer. They provide valuable information about the proper use of existing treatments. They support informed decision-making for both patients and healthcare professionals. They advance medical knowledge and improve patient care. Now that we know why they're important, let's move on to the main uses of clinical trials. Clinical trials have various uses, including treatment efficacy, safety, prevention, and diagnosis. Treatment efficacy. This refers to assessing if a new treatment or intervention is more effective than existing treatments or a placebo in treating a specific condition. For example, a clinical trial might compare a new drug for high blood pressure with an existing one to see if it has fewer side effects. Safety. Clinical trials help determine the safety profile of new treatments, including any potential side effects or risks associated with them. By understanding the safety of a treatment, doctors and patients can make better decisions about its use. Prevention. Clinical trials can also evaluate ways to prevent diseases or conditions from occurring. For example, a trial might test the effectiveness of a new vaccine in preventing a viral infection. Diagnosis. Clinical trials can compare the accuracy of different methods for diagnosing conditions. For example, a trial might compare the performance of a new imaging technique with established ones to see if it's more effective at detecting a particular disease. Next, let's explore the different types of clinical trials. There are several types of clinical trials, each with its unique features and purposes. Randomized controlled trials, RCTs. RCTs are considered the gold standard in clinical research. In an RCT, participants are randomly allocated to different groups, such as a treatment group and a control group. Randomization helps ensure that any differences between groups are due to the intervention rather than other factors. For example, in a trial investigating a new drug for type 2 diabetes, participants would be randomly assigned to either receive the drug or a placebo. This type of trial provides the strongest evidence for or against the effectiveness of an intervention. Observational studies. Unlike RCTs, observational studies do not involve direct manipulation of variables. Researchers observe and collect data without controlling or changing the conditions. Cohort studies and case control studies are common types of observational studies. They are useful for exploring associations between variables and gaining insights into public health issues, but they do not establish cause and effect relationships as strongly as randomized controlled trials. Crossover trials. In a crossover trial, participants receive both the treatment and the control condition. The order of treatment is changed to reduce the impact of any carryover effects. These trials are efficient because each participant acts as their own control, reducing variability. Historical controls trials. These trials compare a new intervention with data from previous studies using standard treatments. Historical control trials are useful when it is not feasible to obtain informed consent from participants to be in a control group or when resources are limited. However, they are susceptible to biases due to differences in patient populations and treatment protocols over time. N-of-1 trials, also known as one-person trials. These trials involve only one person who receives multiple cycles of treatment and control conditions. N-of-1 trials are typically used when there is a need for personalized medicine, like in cases of rare disorders. However, they are limited by their inability to generalize findings beyond the single participant. Let's now delve into some key methods used in clinical trials. These methods are crucial for ensuring the quality and reliability of the trial results. Randomization. As mentioned earlier, randomization is the process of assigning participants to different groups in a clinical trial. It helps to ensure that any observed differences in outcomes are due to the intervention and not other factors. Randomization is especially important in randomized controlled trials. Types of randomization include simple randomization, stratified randomization, and block randomization. Concealment. Allocation concealment refers to keeping the treatment assignment secret until the participant is enrolled. This helps prevent bias in treatment allocation by ensuring that neither the researchers nor the participants know who will receive the treatment and who will receive the control. Blinding. Blinding is used to prevent bias in clinical trials by keeping participants and/or researchers unaware of the treatment being received. Single blinding keeps the participant unaware of the treatment. Double blinding keeps both the participant and the researcher unaware of the treatment. Triple blinding involves all three parties, the participant, researcher, and data analyst unaware of the treatment. Matching. Matching is a technique used in clinical trials to ensure that participants in different groups are similar with respect to specific characteristics or variables. This helps to reduce the impact of these variables on the outcome and enhance the accuracy of the results. For example, in a trial comparing two drugs for treating high blood pressure, participants may be matched based on age, gender, and severity of hypertension. Now that we've covered the key methods, let's move on to the statistical advantages of clinical trials. Understanding the statistical aspects of clinical trials is essential for interpreting their results effectively. Statistical advantages of clinical trials include: 1. Reducing confounding bias. Confounding variables are factors that can influence both the treatment and the outcome. Randomization helps distribute confounding factors evenly across groups, minimizing their impact on the results. 2. Strong causal inference. Clinical trials, particularly randomized controlled trials, provide the strongest evidence for cause and effect relationships. Through randomization, these trials can establish a clear link between the intervention and the observed changes in the outcome. 3. Handling type 1 and type 2. Errors. Type 1 errors are false positives, and type 2 errors are false negatives. Clinical trials are designed to minimize these errors through appropriate sample size calculations, power analysis, and the use of hypothesis testing. 4. Subgroup analysis and longitudinal follow-up. Clinical trials allow for subgroup analyses, which help identify specific populations that may benefit the most from an intervention. Additionally, longitudinal follow-up measures the durability of treatment effects over time, providing a comprehensive understanding of the intervention's impact. In conclusion, clinical trials are an indispensable part of medical research, providing a structured and rigorous approach to evaluate the effectiveness and safety of new treatments, medications, and therapies. From designing experiments to analyzing results, each step plays a vital role in advancing medical knowledge and ultimately improving patient care. Whether it's a groundbreaking drug, a novel surgical technique, or a preventive measure, clinical trials provide the evidence needed to guide medical practice towards the most effective and safe options for patients. So next time you hear about a new treatment or therapy making headlines, remember the journey that brought it to your doctor's office, the countless clinical trials that evaluated its efficacy and safety, ensuring that it reaches patients with the highest standard of quality and reliability. If you found this video informative and helpful, please don't forget to hit that like button and subscribe to our channel for more engaging medical content. Your support means a lot to us and helps us continue creating quality educational content. Also, if you have any questions or comments, feel free to drop them in the box below. We'd love to hear from you and engage with you further. Thanks for watching and stay tuned for more.